Bismillahirrahmanirrahim. In our previous lectures, we discussed the mechanism of cell injury, in which we discussed the membrane damage, mitochondrial damage, influx of calcium, and the accumulation of reactive oxygen species in the cell, and the accumulation of misfolded proteins and damage to the DNA. And all these processes lead to cell death or necrosis. Now, today we are going to discuss uh, a few important examples of cell injury, and we will discuss the ischemic and hypoxic injury. Then we will discuss the ischemia-reperfusion injury, which is very important, particularly in cases of strokes, myocardial infarction, organ transplants. And finally, we will discuss the injury that is caused by chemicals and toxins as well as medications. Now, there is a difference between ischemia and hypoxia. By ischemia, we mean the reduced or diminished blood supply to a tissue, while in hypoxia, there is decreased oxygen level. There is a deficiency of the oxygen. So we can say that the major cause of hypoxia is ischemia, because when there is reduced blood flow to a tissue, the tissue suffers from hypoxia because there is deficiency of the supply of the oxygen. However, hypoxia can also be caused without ischemia. There will be no ischemia, but there will be hypoxia. And in those conditions, what happens that the blood supply is normal, but the blood doesn't contain enough oxygen. And this poor oxygenation of the blood or hemoglobin may be seen in conditions like anemia, in which there is a deficiency of red blood cells, and also in conditions where a person is at high altitude, like decreased partial pressure of the oxygen. And additionally, different lung diseases, for example, the obstructive lung diseases and restrictive lung diseases, lead to poor oxygenation of the blood. The transfer of oxygen to the blood and the transfer of carbon dioxide from the blood to the air that is to be expired, it is damaged. And the result is that there is poor oxygenation of the blood. Now, in this condition, we can see that there is no ischemia. The blood supply to the tissue is normal, but the blood itself is deficient of the oxygen level. And this deficiency may be caused by anemia in conditions of decreased partial pressure of the oxygen, and in many lung diseases in which there is poor oxygenation of the blood or hemoglobin. Now, as I said that hypoxia, the major cause of hypoxia can be ischemia, while hypoxia can also occur in the absence of ischemia in conditions that we discuss. Now, what happens next? That whenever there is hypoxia, what happens that there is a deficiency of the oxygen, and the process of oxidative phosphorylation for the generation of ATP in the mitochondria, it is disturbed or damaged, and as a result there is little ATP formation. Now, what happens if in simple hypoxia, that since in hypoxia the blood supply, arterial supply and venous drainage, they are continue, and the alternate way of ATP generation, that is known as the anaerobic glycolysis, in which the glucose is utilized in the absence of oxygen for the generation of ATP. So in conditions of hypoxia, the anaerobic glycolysis continues for some time, while on the other hand, in hypoxia that is caused by ischemia, the anaerobic glycolysis or the alternate pathway of the ATP generation also becomes compromised. We know that in the absence of oxygen, glucose from the blood or glucose that is derived from the breakdown of glycogen known as glycogenolysis is used for the generation of ATP. And we know that this process is short-lived, but the cells which have enough supply of the glycogen can maintain the process comparatively for a longer time than when we compare it to the hypoxia that is caused by ischemia. So why it so happens that in ischemia there is reduced delivery of substrates like glucose for glycolysis. As a result, the glycolysis, the anaerobic glycolysis is very short-lived. Moreover, the cells, they utilize their glycogen which is converted into glucose and used for glycolysis, and this will lead to a rapid depletion of the glycogen stores in the cells. And the third very important point is that in hypoxia without ischemia, the blood supply continues, and as a result, the metabolites that result in chemical processes and cellular metabolism, they are removed by the venous blood flow. But in ischemia, what happens that the venous blood flow is also compromised, and as a result there is decreased removal of the metabolites, and the accumulation of lactic acid as a result that occurs in anaerobic glycolysis will decrease the pH inside the cell, and it will inhibit the anaerobic glycolysis. Now, ischemia can be caused by many conditions like atherosclerosis, in which there is atherosclerotic narrowing of the blood vessels due to deposition of the cholesterol, particularly the LDL cholesterol. It can also be caused by thrombosis, in which there is a thrombus formation, clot formation within a blood vessel, and sometimes if this thrombus is detached and carried in the circulation to somewhere else, known as the embolism, it will also cause ischemia. And sometimes pressure from the outside, for example by a tumor, may compress the blood vessel and will result in decreased blood supply or ischemia. And in conditions, generalized conditions like hypotension caused by the shock, will also lead to a deficient supply of oxygen to the tissue, and as a result, what will happen that the oxidative phosphorylation, which is the electron transport chain and that occurs in the mitochondria for the generation of ATP, it becomes defective. And the result of decreased oxidative phosphorylation, there will be decreased generation of the ATP. And we know that ATP is needed by the many cellular processes that are energy dependent or ATP dependent. So the one very important is the sodium potassium ATPase pump. We know that in sodium potassium ATPase pump, which is an ATP dependent or energy dependent membrane pump, what happens that the three sodium ions are they are pushed to the outside of the cell to the extracellular space, and in return two potassium ions that enter the inside of the cell, and the sodium potassium ATPase pump maintains the equilibrium, the ionic equilibrium within a cell. As a result, what will happen that there will be increased entry of sodium, and we know that this entry of sodium will be followed by the entry of water, and this situation will lead to swelling of the cell. Accumulation of sodium followed by water will lead to the swelling of the cell, and also there will be increased efflux of the potassium from the cell to the extracellular space. Additionally, the calcium transport system, the calcium transporters, they are also deranged because of the deficient ATP, and these processes also are energy dependent processes, and they depend on the adequate supply of the ATP. So what will happen that the entry of sodium and water will cause endoplasmic reticular swelling, and as a result the plasma membrane microvilli are lost and there are the membrane blips formation. And we know that the entry of calcium will activate many proteolytic and hydrolytic enzymes. Now the second component that occurs as a result of deficient ATP is the anaerobic glycolysis. And we know that anaerobic glycolysis, which is a short-lived process, and at least some ATP is provided to the cell for the metabolic processes. But as I said, this is a short-lived process, and what will happen that the glycogen stores, they will get depleted because the glycogen is converted into glucose through glycogenolysis, and as a result the glycogen stores, they are depleted. In the anaerobic glycolysis, we know that there is production of the lactic acid, and this lactic acid will reduce the pH of the intracellular compartment. And what will happen that the acidic pH in the cell, it will cause inactivation of not only structural proteins but also the enzymes, and they will stop functioning, and as a result the cell becomes damaged. The deficiency of ATP or decreased ATP through oxidative phosphorylation in the mitochondria, it will lead to the detachment of ribosomes, and we know that the ribosomes that are attached to the endoplasmic reticulum, they are the sites for protein synthesis. So as a result the polysomes are converted into monosomes, and this detachment of ribosomes from the endoplasmic reticulum will lead to the decreased protein synthesis, which is the major function of the ribosomes. Now in this slide we can see that we have studied that the cell initially passes through a stage of reversible cell injury, and in reversible cell injury, if the cause is removed, the cause of cell injury is removed, the cell has a potential to become normal again. And we know that hypoxia, especially hypoxia caused by ischemia, what will happen that if it continues and if the damaging agents are not removed, what will happen ultimately that the swelling of the endoplasmic reticulum and detachment of ribosomes from the endoplasmic reticulum, the process is further exaggerated if the injurious agent is not removed, if the blood supply is not restored. And what will happen that the lysosomes in the cell that contain various hydrolytic enzymes enclosed in the lysosomes, when the lysosomal membranes are damaged, they will rupture, and these proteolytic enzymes are released into the cytoplasm of the cell, and they will cause degradation of the cell organelles. Now if the injury continues, what will happen that the DNA and nuclear materials are damaged and there is nuclear condensation. And we have discussed in our previous lecture that degradation of the nucleus may occur through the process of karyolysis, karyorrhexis, and pyknosis. Now what happens that the damage mitochondrial membranes and other membranes of the cell organelles. Finally, the cells are replaced by the myelin figures. And what are these myelin figures? The myelin figures, these are whorled masses of phospholipids, and sometimes calcium may be deposited in these whorled masses. And as a result of all these changes, the cell membrane and nucleus will undergo fragmentation, the membrane fragments, nucleus fragmentation, which is known as the pyknosis. Now these cells finally appear in the form of these dead cells. They form the myelin figures, and they appear under the microscope in the form of myelin figures, which I said are whorled masses of the phospholipids. Now these myelin figures are either phagocytosed by the infiltrating leukocytes and removed, and sometimes if there is excess formation of the myelin figures, what happens that the triglycerides contained in these masses, it is converted into fatty acids broken down into fatty acids, and if calcium is deposited in these fatty acids, the process known as saponification of calcium soap formation will occur. Now we go to the second example of the cell injury, and that is the ischemia-reperfusion injury. So if a tissue is deprived of the blood, if there is a deficient blood supply to a tissue, and if this ischemia causes ischemia hypoxia induced damage, but if we restore the tissue, or if we restore the blood supply known as the reperfusion, so what we expect that normally this reperfusion should make the cell recover. However, in certain situations, particularly in conditions like myocardial infarction and cerebral strokes, what happens that when the blood supply returns to the tissue after a period of ischemia or lack of oxygen, anoxia and hypoxia, this reperfusion and restoration of the blood supply will cause further damage, further more damage than caused by the initial insult. And what happens that in reperfusion injury, the restoration of blood flow to the ischemic tissue, which is however viable, irreversibly injured, it will result as unexpected in the more and exaggerated death of the reversibly injured cells and increasing the cell damage. Now, reperfusion injury is particularly very important in conditions of myocardial infarction, in which there is ischemic necrosis of the cardiac myocytes, in conditions of stroke, cerebral stroke, and in conditions of bypass surgery, in which the blood supply is surgically re-established to a necrotic or ischemic tissue, and in conditions of organ transplant like kidney transplant and other organ transplant, and also in conditions where there is hemorrhage, and if it is followed by the resuscitation or restoration of the blood supply. Now, what happens when there is ischemia to a tissue? It will result either, if the ischemia is severe, it will result in irreversible injury to the cells, and the cells, they undergo necrotic or sometimes apoptic cell death, and now these cells are lost and we cannot reverse these dead cells. But some cells, they are injured, but they are in a stage of reversible injury. By that we mean that if we restore the blood supply, these cells can recover and they can become normal again. However, in addition to the irreversibly lost cells and to those cells that are completely recovered after restoration of the blood supply, an additional component of the reperfusion is that many cells that are reversibly injured, and if they do not fully recover, what will happen that the reperfusion will cause additional cell death by necrotic or apoptic cell death. And here this is something paradoxical. By that we mean that we expect that the blood supply will restore all the cells and there will be a recovery, but unfortunately sometimes this reperfusion results in additional cell death of those reversibly injured cells. And this type of injury is known as the reperfusion injury, and it is particularly seen in conditions of myocardial infarctions, cerebral strokes, and acute renal failure. Now, let us have a brief insight into the reperfusion injury. So, what happens in the reperfusion injury, that the two major determinants that are responsible for the reperfusion, ischemia-reperfusion injury are the accumulation of the radioactive species. And the second important component is the inflammation that is induced by the ischemia. What happens that when there is ischemia, loss or decreased blood supply to parenchymal and endothelial cells, what happens that the mitochondria in these cells become damaged. And as we know that the mitochondria are the major uh, sources of ATP generation and in ATP production in the form of the currency or energy of the cell, what will happen that as we know that ATP generation occurs through partial reduction of the oxygen in the electron transport system. But when there is partial reduction of oxygen in which first an electron is added to oxygen and it is converted into superoxide ion, then addition of other electron later to be converted into hydrogen peroxide, and then addition of 3 electrons will lead to the production of hydroxyl radicals, and superoxide, hydrogen peroxide and hydroxyl ion, they are very strong and damaging free radicals. Finally, the addition of 4 electrons to the oxygen will lead to the formation of water, which is innocent. But in this condition, we can see that the reduction of oxygen is not complete. And this partial reduction of oxygen will lead to the formation of superoxide, hydrogen peroxide, and hydroxyl radicals. When the mitochondria of the cell, particularly the parenchymal and endothelial cells, if it is already damaged by ischemia, so there will be increased in the incomplete reduction of oxygen. In normal mitochondria, the reduction of oxygen is comparatively not so much deficient as that what happens in damaged mitochondria. In damaged mitochondria, there will be excessive and exaggerated incomplete reduction of oxygen, and it will lead to the formation of the reactive oxygen species. Moreover, these cells contain enzymes like oxidases, for example, NADPH oxidase, that will also lead to the formation of the free radicals. Now, what happens when the blood supply to these cells is re-established or reperfused? There is an excess supply of oxygen, and as a result, the excess formation of the reactive oxygen species, and there is infiltration of the leukocytes by the inflammation that is induced by ischemia, and we know that these leukocytes contain in their lysosomes various hydrolytic enzymes and can cause further damage. Now, in the face of damaged mitochondria and the excess supply of oxygen and infiltrative leukocytes that happens in reperfusion, what happens here that the cellular antioxidant mechanisms, they are also made compromised by the ischemia. So, for example, the catalase enzyme, glutathione, superoxide dismutase, which are the cellular antioxidant mechanisms in damaged mitochondria, they are also compromised. As a result, the balance tilts in favor of the reduction tilts in favor of the production of the reactive oxygen species as compared to antioxidant mechanisms, and this will lead to the accumulation of the free radicals, and it has got a very important role. The reactive oxygen species has got very important role in ischemia-reperfusion injury. Now, the second component that is mainly responsible for ischemia-reperfusion injury is the induced inflammation by the ischemia. When the inflamed tissue is reperfused, when the blood supply is restored, what happens that lot of leukocytes come in the new supply of blood along with the various plasma proteins. Moreover, there is an influx of calcium to the cells. And the third very important component is the complement activation. Complement activation we will study when we will study inflammation, and complement activation are the complement proteins are important mediators of inflammation, and we will study that complement consists of 9 proteins from C1 to C9, and they have got very important role in giving protection to the cells and tissue. But potentially, if this complement is inappropriately activated of activated, or if it's the activation is exaggerated activation of the complement system, potentially it can cause the damage to the healthy tissues, and as a result there is exaggerated cell injury than the initial insult, which is caused by the inflammatory reaction that is set on. Now, coming to the summary of the reperfusion injury, we have said that the initial ischemia will damage the tissue. When the blood supply is restored, either the reversibly injured cells, they will undergo recovery to normal, and the initial ischemia, if it has got in the loss of cells, those cells that are irreversibly injured, they are permanently lost. And these necrotic cells are cells that die by apoptic cell death. They have no way to return. Now, in those cells that are reversibly injured, a portion of the cells they may recover, but many cells that are initially reversibly injured by the initial ischemia, they will undergo cell death when the blood supply is re-established and the tissue is reperfused. These conditions are seen in myocardial infarction, stroke, and acute renal failure. Now, the two important components that we discussed is the inflammation. Now, when there is ischemia, it will induce inflammation, and when the blood supply is restored, there will be infiltration of more and more inflammatory cells. And why it is so? The parenchymal and endothelial cells in the ischemic portion of the tissue, they express various cytokines and adhesion molecules that will lead to the infiltration and recruitment of the neutrophils and other leukocytes. So inflammation is an important component of ischemia-reperfusion injury. The other important component is the increased generation of the reactive oxygen species. Now, these reactive oxygen species are produced by parenchymal cells, endothelial cells, and also the new leukocytes that are recruited to the area of inflammation. As we said that the incomplete reduction of oxygen in the mitochondria will lead to the increased production of the reactive oxygen species, and the various enzymes, oxidases and hydrolysis present in these cells, when they are released, they will degrade the cell organelles and will cause severe damage. In the case of necrosis, there is also release of cellular contents into the extracellular spaces, which may induce inflammation. And the one important component of that contribute to the ischemia-reperfusion injury is that the initial ischemia will cause compromised antioxidant mechanisms that we discussed in our previous slide, and as a result, the balance tilt in favor of the production of and accumulation of the antioxidant mechanisms. Complement activation, which consists of various complement proteins known from C1 to C9, and that also contain the membrane attack complex, is an important part of inflammation, and the complement proteins are important mediators of inflammation. And in ischemia-reperfusion injury, there is activation of these complement proteins, and as we know that in addition to giving protection to the tissue, the complement proteins have also a potential to cause severe damage to the healthy tissues. Now, The third type of injury that we are going to discuss today is the cell injury that is caused by chemicals and by toxins and by various medications. Now, there are two mechanisms. Either these toxins, chemicals, or drugs may bind directly to a cell component. For example, in conditions of mercuric chloride poisoning, what happens that the mercury binds to the cell membrane proteins, and the same effect we use by giving the antineoplastic drugs to kill cancer cells. Now, what will happen that when these molecules bind to the cell membrane proteins, they will induce inhibition of the ATP dependent membrane transport or sodium potassium ATPase pump and calcium transporters. And as a result, when the membranes of these cells is damaged by these chemicals or toxins, it will lead to cell damage and cell death. Now, the second mechanism in which the chemicals, toxins, or certain drugs they cause cell damage is that initially these toxins are not cell damaging, but they are converted to various toxic metabolites which cause severe cell damage. For example, the acetaminophen, paracetamol that causes liver damage, and the carbon tetrachloride. Now, here we can see that the carbon tetrachloride is converted by the P4 oxidase system. It is converted into a potent free radical known as the trichloromethyl radical. Now, this CCl3 or trichloromethyl radical is a very toxic free radical, and it can damage the membranes of the cell and organelles by doing phospholipid peroxidation. We know that the free radicals they damage the membranes by lipid peroxidation. They also can do modified oxidation of the structural and functional proteins, and they can also cause damage to the cell nucleus. So at the end, what happens that there is breakdown of endoplasmic reticular membrane. So what happens if the process which mainly runs in the liver, there will be decline in hepatic protein synthesis. There will be dissociation of the ribosomes from the smooth endoplasmic reticulum, and as a result there will be decreased synthesis of the apoproteins. And we know that the apoproteins are very important in lipid carrying proteins known as the lipoproteins. The examples are high density lipoproteins, HDL, LDL, and intermediate density lipoproteins. So what happens that the dissociation of the ribosomes from the smooth endoplasmic reticulum will lead to the decreased formation of the apoprotein and lipoproteins. And as a result, the lipids are not properly carried by the lipoproteins outside the liver, and as a result there will be accumulation of these fats in the liver known as the fatty liver of carbon tetrachloride poisoning. Thank you very much.